So good morning, everybody. I think you might have already just heard me say um, there was a story that came out on public radio almost a year ago now, talking about the work that our speakers are going to talk about this morning, and it was really interesting. And I thought, it would be great to hear more about the work that they're doing. In Duluth, they've been doing some intensive stream restoration projects as a result of the flooding and the serious damage caused there, and other human activities that have disrupted the streams. And the goal of these projects have been not only to repair the channel damage, but also to restore and improve the habitat for fish and other aquatic species. So our panel of speakers today, Valerie, Brady, Karen Gran, and Brad Everett, have been working on evaluating the success of these restoration projects by measuring fish and macroinvertebrate habitat and the stream channel stability and connectivity and so on. This morning, they're going to tell us what they have discovered in their research. So just quickly introducing our speakers this morning. Valerie Brady is interim leader of the Natural Resources Research Institute Water Research Group. I think I have that right, Valerie. Her research focuses on tracking the condition of streams, wetlands, and Great Lakes coastal areas using indicators of biotic condition and assessing the success of stream and wetland restoration projects, investigating invasive species impacts on habitats, ecosystems, and food webs, and investigating stormwater runoff and land use effects on aquatic ecosystems. Karen Gran is a professor in the Department of Earth and Environmental Sciences at the University of Minnesota Duluth. Her research into rivers has taken her from Minnesota to the Philippines and back again. That's very interesting. And for the last decade, she has been working to understand how rivers within Minnesota have evolved since the last glaciation and how we can use that information to better manage them. And Brad Everett has just recently started at the MPCA as a hydrologist in solid waste permitting at the MPCA there in Duluth. Prior to starting at the MPCA, he worked as a hydrologist in remediation at the Wisconsin DNR. He moved to Duluth in 2021 to get a master's degree in water resources, and that was also at the University of Minnesota at Duluth. He also worked for several years as a subsurface petroleum geologist for ConocoPhillips and PDC Energy in Houston and Denver. It is great to have you guys with us to talk about this really interesting work. And so, Valerie, I'm going to yield the floor to you. You can take it and share your screen and take off with it.
Okay, and I believe I am showing you the slideshow.
Yes, we can see it.
Okay, perfect. All right, so just first a really quick introduction to the Natural Resources Research Institute, since not everybody knows about that, even though most people are familiar with the University of Minnesota Duluth. And so we are in, oops, both Duluth, it changed slides, right? Okay, we are in both Duluth and Coleraine. We employ about 140 people at any given time, and we do all sorts of research projects, all focused on trying to help improve Minnesota's both economy and environment. And so we do all sorts of stuff, ecology projects, wildlife, forestry, and our Coleraine lab, which is an old former U.S. Steel research facility, looks at better ways to work with our mineral resources in Minnesota with fewer impacts on the environment. And that's where the groups like mine come in, is looking at those environmental impacts. So we can cover all of that. And as I said, the whole idea is to have a future economy for Minnesota that is strong, but is also the least detrimental to the environment that we can make it. We're often hiring a person or two. So if you know anybody who's looking for a job in these areas, please do send them our way. And our whole goal is to be integrated in these projects. And so we're looking at not only mining activities, but ways to clean up the sulfate and reduce the sulfate impacts on the environment and reduce the sulfate in the waters that come from mining activities, as just one of many, many examples. So speaking of water, and we had a great introduction today. So you already know a bit of background about why we wanted to do this particular project, which we got funding from LCCMR to do, the Environment and Natural Resources Trust Fund. So with the floods and the other damage and things that have happened to streams, you might know Duluth's topography is a little bit steep, and so it can get some real damage to the streams there with these really higher flows that we are getting from climate change. We wanted to take a look at what these realignments are doing. So, a whole channel realignment where they re-meander the stream is a fairly intensive bit of restoration work for a stream channel to endure. And so with all of these going on, this gave us the opportunity to go in and look at a whole bunch of things. And we have a whole bunch of people, you can see them on the screen, who are working with us and collaborating with us on the project because we are trying to tackle so many different aspects of stream ecology in this process. And so we want to know, first of all, of course, are these realignments meeting their goals and holding up over time as we get more of these intense stream flows? And what are the effects on not just the goals and the targets, often fish habitat, improved invertebrate habitat, our goals, but can we look even deeper and assess stream ecosystem function in ways that, because of the complexity and the cost and the time that they take, typically can't be done just as part of the restoration project itself. And so we wanted to do a much more comprehensive look, take a look at, stream habitat, fish habitat, how the bugs are doing, but also this more in-depth look all at the same time. And so now, as I said, fish and invertebrates, but also Karen's specialty is looking at geomorphic stability. We're both looking at habitat in different ways. And we threw in nutrient cycling, something that often we don't have time to do in stream restoration assessments. So again, how does the stream process the nutrients and how quickly are those used up and recycled going downstream? And then what we're going to be presenting mostly on today is this connectivity with the hyporheic zone. Because you think of going in with heavy machinery and re-meandering a stream channel, it is possible that we could either increase or decrease connectivity with the hyporheic zone and the water just under the stream bed, that connection to the superficial groundwater, which is pretty important, especially for the bugs and fish in the streams, and also for keeping many of these streams cooler as we get into climate change. So to do this comparison, we're looking at the realigned reaches and also control reaches, which are typically upstream in the same stream. Now, ideally, we'd want to do this with a Baki, which is looking at each reach, the control and the realigned reach, both before and after there was realignment. Um, but projects are not long enough typically to do that. We just can't find that level of funding. You'd need like 10 years. And so we are able to do it on 2 of the streams we're studying, and we'll be sampling them for the after realignment part this coming summer. But unfortunately, for most of them, we're coming in after the realignment has been done. But we still have the control, the unrestored reaches upstream of the treatment. One thing to note, control does not mean pristine. There aren't too many of those left these days, especially in Duluth. It just means that they weren't realigned. Um, and control is not the same thing as the geomorphic reference that was used to design the channel realignment. Control, we wanted them to be in the same system, right upstream, same fish, bug habitat, that type of thing. So our site locations. And this slide shows you the control and the treatment and what we're, just a little bit what we're doing there. So we're going to be talking most about Mission Creek today as an example. And that is where the radio story that Mark was talking about was done. And so that's what Brad's going to be presenting on. We also have in the Duluth area, Sargent Creek, Chester Creek, which is a bit unique in this study in that it includes not only a control, but a degraded control that has been damaged but has not been realigned. Keene Creek is where, and Miller Creek are where we were able to go in before channel realignment and get some sampling before that was done. Um, Up the shore, we were trying to look at East Beaver, and East Beaver's realignment and restoration work got taken out in the 2022 flood, which was kind of sad. You can see East Beaver is here labeled number 3 in this slide. I can tell you with absolute certainty that it does not look like this anymore. That one did not hold up, but we're doing a bit of a post-mortem on that one to learn what we can from these ones that don't hold up. Mission Creek is picture number 2. And in fact, I think this picture was taken when we were out there doing the dye study that Brad's going to be talking about. So I'm going to go through methods really quickly, and but Brad is going to do an in-depth on the methods for his part of the study. So we're doing fish sampling. Not only are we doing electrofishing, we're also looking at doing snorkel surveys to see if brook trout are actually building their nests, their redds, in the realigned and control streams. Habitat assessment, of course. Fish need to eat, so looking at the macroinvertebrate samples, the macroinvertebrates in the streams. And I think, Karen, this is where you take over.
Yeah, I'll talk a little bit about some of the geomorphic assessments and the functional assessments that our group is working on. On the geomorphic assessment side, some of it is just doing some change detection, looking at erosion and deposition. This is work that's starting up this summer. So we'll be doing drone flights to get topography using structure from motion, kind of on the banks and floodplains, and coupling that with total station surveys in the channel that align with some of the habitat surveys that I'll talk about in just a sec. And then we can compare these topographic data with plans or as-built surveys on those treatment sites. We don't have the same kind of information on the control sites. We do have LiDAR data. And so part of this is assessing to see what kind of information we could get from different sampling methods. Next slide. And then we're also doing some comparative surveys. We are doing physical habitat surveys usually using the EPA's NRSA standards. So we get detailed bond profiles, grain size distributions, cross sections, riparian vegetation cover, et cetera. And we're also measuring metrics in the Minnesota SQT, or stream quantification tool. And the idea is that that is intended to be a measure of functional, kind of a functional assessment of the stream. And so we can see how the streams show up in the MN SQT, and then see how the rest of the data that we're collecting compare with that. I did want to mention that the Soil Water Conservation District is doing the similar analyses with reference reaches. So these are the design reference reaches, sort of before and after a similar length of time. So we can see how much change occurred there and compare it with the same methods to our data. Now I can go to the next slide. So I just want to say a few words about the SQT for those who aren't as familiar with it. This is relatively new. We're using kind of a modified version of the SQT. It's built on the Stream Functions Pyramid by Will Harmon and is designed to provide a method to quantify sort of ecological integrity with specific measured metrics. You can go to the next slide. This is the slide that might be hard to read on WebEx here, but there are these 5 functional categories of hydrology, hydraulics, geomorphology, physicochemical, and biology. And the idea is that you could measure a site, calculate how it's functioning, and then look at the restored site and see what the functional uplift is. So this is something I think we're going to be hearing more and more about as it moves into policy in the state. And so we wanted to be able to compare how these were working with other ways to measure functional ecological function. So next slide. And then some of the functional assessments that we're doing, we will be installing stream temperature loggers. That's part of the work that Val's group is doing. We have run a bunch of nutrient uptake tracer studies using the task method, and these are very similar. Well, they're the same methods that the EPA has been using for many years. With Terry Jica and Brian Hill have done that. And so, and I just pronounced your name wrong, Janke. And so we're doing the same methods on both the control and treatment sites to compare that. And this is work that David Baldus did for his thesis. And then lastly, the hyperic exchange tracer study. And I will stop talking now, and I'll pass it off to Brad to talk more about that specifically.
All right, so hyporheic exchange, for those of you who may be unfamiliar with the term, a hyporheic zone is generally the area right around underneath the stream bed and to the sides on the banks, where surface water from the creek flows into that zone of the subsurface and then returns to the stream at some point. Um, these are kind of nested flow paths. Some are very long, some are short. The study that we're going to talk about today is really going to focus on these shorter flow paths, kind of within a reach, and these are typically through riffles, maybe gravel bars, a little bit, but it's got to be a flow path that is short enough to enter the subsurface and return to the subsurface during the duration we're sampling. So that's kind of a limitation on what we're looking at. But past studies have shown that the bulk of the exchange for the surface water in the hyporheic zone occurs along these shorter flow paths. Next slide. So the method we're using is this reactive tracer dye tracer called bezazerin, which converts to this compound called resirufrin. And the way this dye works, it's been around for a long time. It started in the 1940s in the dairy industry to look for microbial or bacteria activity in milk. And what this bezazerin does is in the presence of aerobic metabolism, oxygen consumption, it an oxygen gets stripped off the bezazerin, which converts it to resirufrin. They're both fluorescent and they fluoresce at different wavelengths, so you can measure them quite easily in the lab. Past studies have shown that all that transformation in a stream system occurs in the shallow sediment, where the microbes live on the sediment surfaces. And in the stream column, the water column itself, there's very little to no transformation. There's just not enough microbes in the flowing water to convert it. And so we use that conversion amount, how much of the decay of bezazerin as a proxy for hyporheic exchange, because you can't, it's not really going to convert unless that water carrying the tracer has entered the subsurface, been exposed to those microbes, and then returns to the stream. And again, we're really only working at flow paths on the order of several hours at the most, because anything that enters those longer flow paths won't return during the time that we're sampling. And so one thing we kind of had a hypothesis going in, you heard Val mention at the beginning, when they're doing the earthwork to realign these streams, there's a lot of digging going on. They're laying sediment on top of whatever the underlying geology is. In Duluth and a lot of places that can be clay. And so the thinking was is that they're compacting that clay and they're digging a channel within that clay. There's probably going to be less exchange was the initial hypothesis going in. In the realigned reaches, I was expecting to see lower processing rates and therefore lower hyporheic exchange in the restored reaches. Next slide. So the way we did this, and this is kind of what they were talking about on the radio program, on the radio story, was we take the bezazerin and we mix it in a bucket. It's pretty low tech. This is what, when we're talking about, Val was talking about the treatment reaches and the control reaches. This is the stream, the treatment's downstream. We have our injection points and our sampling points. I'll stop pointing with my mouse because that doesn't do anything. We get out there, the first thing we do is we measure discharge. And then there's a program that was developed by our collaborator at the University of New Mexico to enter the discharge and some other stream parameters to get a sense of how much tracer to use, both the conservative tracer and the bezazerin. And it does not take much bezazerin. It's a very strong dye. When you see the pictures coming up, I think the most we ever injected on a stream was like 10 grams. So it's not much at all. As opposed to we used salt in 2022 as a conservative tracer. I'll discuss that more later. We were putting up to a kilogram of salt in these for sampling over 100 to 200 meter reaches. Collect an ambient sample so you know what the background sodium or the chloride concentration is. the nice thing about Resazurin as a tracer is it doesn't exist in the stream naturally. There is no background, so anything you detect is because you put it there. You mix these tracers in the bucket. Resazurin takes some serious stirring. It's not nearly as soluble as salts, so it takes a little while, and it blows around. You got to be careful with it so it doesn't get in the stream before you want it to. And then the way we sample at the top and bottom of these reaches, we have a connectivity meters deployed at each sampling site. We sample when we start noticing the connectivity change from the conservative tracer that we added, sample through until it returns to ambient, and then we take a sample half hour, it returns to ambient or back where we started, and then another sample an hour after that to make sure everything has gone through and we're back to where we started. All right, so next slide. And so this is what it looks like. So this picture with the bright pink purple tendrils moving downstream, that's what all of 10 grams mixed in a 5-gallon bucket look like when you dump it in the creek. So it's very vibrant. It looks really cool. You can watch it go downstream. The next image immediately to the left of that is you can see to the right of that riffle the clear water where I injected, and then you can see how purple it is after that. On the far left is an injection site at the end of the day. Afterwards, you can see a little bit left there and some of the macrophytes there, or the algae on the bottom. So some of it stays kind of trapped in places. That's transient storage. And then on the far right, that's what our sampling system looks like. It's pretty simple. We sample with a syringe, we filter, and then we just inject it into plastic bottles and put it on ice until we get back to the lab, and then we put it in the fridge or the freezer, depending on how long until we're going to analyze, because you don't want the microbes that happen, the small amount of microbes in the bottle to continue respiration and conversion. So you want to stop that. All right. So then we take it into the lab for the measurements on the Resazurin and Resarufrin. I mentioned that they're fluorescent, so it's pretty simple. Put them in the spectrophotometer, and then you get a fluorescence intensity. You make standards. You relate intensity to concentration. The conservative tracer was something we learned a lot about. For this study and previous studies and nutrient studies, they use a colorimetric. So they add chemicals to your sample, they change the color that's related to how much chloride you put in there, or how much chloride's in the sample. I'm adding a dye to my samples, which makes them blue, which changes the color, which can impact the chloride results. I want to note that I'll talk about Keene Creek data a little bit later. That was the first creek we did in summer. Our Resazurin concentrations were quite low, so the samples didn't look dyed at all, and we ran those with the colorimetric method. So then that was the only chloride data we got from those. We think the chloride data is probably good, but there's that caveat there. And then the next stream we did, the samples were blue. I think if you go to the next slide, it'll show it. Oh, no, it comes up later. The samples were blue, and the lab calls and says, why are your samples blue? That's a problem. So I'll discuss more about the conservative tracer learnings in a little bit. So we gather the data, the concentrations from the lab, and then now we go to the next slide, and you get these are called breakthrough curves on the right, and that's just concentration through time in your samples. Using the area under those curves, and then also the center of mass for the time of those curves, you get how much mass moved through, how much tracer moved through your upper site, or how much mass there was, and then how much mass moved through the downstream site, and the time comes from the center mass of those curves, essentially. That's what tau is. And then you're just relating how much decay in Resazurin was there over time, and that's what that bottom equation is. That lambda 1 is the bulk processing rate. So it's a pretty simple algebraic equation that was derived by our collaborator at New Mexico. Other things people have tried is you can use like full transient storage modeling with this data, trying to match your observed data to get some more parameters. Um, I attempted to do that, and the collaborator at New Mexico said that it's very sensitive to the your initial guesses that you put on all the parameters in the transient storage model. And so he felt that this bulk Razazaran processing rate was uh, a better proxy or a more um, less uncertain process proxy for hypereik exchange than going through the full transient storage modeling. So that's the result we're looking at in my data coming up is bulk Razazaran processing rates. And again, that's just the decay in the Razazaran from the upstream site to the downstream site over our reach of interest. And then how fast that occurs. And a higher number, bigger number equals more exchange is the assumption that we're making. Next slide. So again, blue samples there in the top right, and all the fun things we learned about using sodium chloride as the conservative tracer. We needed to switch from that colorimetric method to using an ion chromatograph, which increased some costs and time. And it just, then when we actually used the ion chromatograph, we had a high number of samples. I had over 600 samples from the creeks that we had done. So we ran them in batches, and there's a lot of samples, so several people were helping getting their samples prepped. And we just noticed that sometimes the same creek, the same sampling site, if not all the samples were run in the same batch, we would see offsets in the chloride concentration data. So that was it's really challenging to use that in the lab. And so just something to note. And then the last thing is that some of our streams have, no surprise, with all the road salt, very high background chloride data. And that's what this bottom graph is showing on the right. That's the change in chloride over 24 hours at the Chester treatment reach. And so you can see, at the end of the day, we went up 5 milligrams per liter, but after 24 hours, it was 10 to 15 milligrams per liter higher on the sampling sites, which is a lot because our conservative tracer is supposed to come back down to where we started. And so this just adds some uncertainty in that center of mass calculation on those breakthrough curves and the time it takes, which impacts the Razazaran processing rate. If these are going to be skewed later or anything like that, that just impacts that. So that adds some uncertainty to our data. It's kind of unquantified uncertainty to a degree. So like this one on the right, you can just see how that's basically we're attributing that to evapotranspiration over the 24 hours. And one thing that's interesting, you can see in that table there, some of these, Mission Creek has had the highest ambient chloride concentrations, and it doesn't really correlate with the urban density there, percent urban development in the watershed. So that's one thing that's interesting with Mission Creek, and so that's why that's the site that we talked about at the end that the radio story was on, and that's why we went back out in 2023, which I'll get back to in a little bit. So the next slide, we talk about what I did with the conservative tracer data. I wanted to get something out of all the data we collected, so I had to figure out how to make some of this data usable. So some streams like Sargent, it had the lowest ambient chloride concentrations. Most everything got run in the same batch. So the top lines, those are the conductivity. The green and red are conductivity. The blue and brown are the measured chloride concentrations, or changing chloride concentrations, excuse me. And you can see that they look pretty smooth. The chloride curves look pretty similar to the field conductivity curves. The bottom graph is emission with the really high values, and you can see that a major offset from the different day in dilution in the brown. You can see very little response at all in the blue curve, and it's noisy. So what I ended up doing is I went by site and looked at the raw conductivity, field measured conductivity that was recorded during when each sample was taken, and then made these linear regressions to then convert my measured conductivity from the field into chloride concentrations with data that was quote unquote trustworthy before a change, an offset due to a different batch being run. So looking at the data, look which one had the most data from that site in the regression, and then converting the offset data up to that regression. Okay, so then that's what I use to calculate the time. So then the results of the processing. Yep. The main observation is that there's really no significant difference between control and treatment reaches. Some creeks, the treatment had higher processing. Some of them, they had lower the treatment had lower processing. So again, starting with Keene on the far left, as Val mentioned, that's the one we did before any restoration. So those are both of those are in their quote unquote unrestored states. And the control in this instance had a higher rate. You'll see that everything else is, and it's actually in a higher order of magnitude. The discharge at Keene was the first creek we did in the season, so it was June, and the discharges were over 100 liters per second, I think is what I did everything in, where the other ones are 10 or less. And so discharge does correlate to, or it does scale with hyperreac exchange. Higher discharge, there's higher energy to drive that flow. And then the three in the red box, those are kind of the three that I have good data on. They're both before, they both have restored reaches and control reaches. And then Miller on the far right, the data from the treatment reach wasn't sufficient to get a processing rate. So I didn't actually able to get, and that would have been a pre-treatment. So I don't have that there. I just have the control processing rates. So you can see at Sargent, the treatment has higher processing rate. The Chester, it's the control. Mission, it's the treatment. And this is all from the 2022 data with the sodium chloride tracers. So a couple things that are interesting. The Miller control is a wetland stream that has like almost no riffles in it. I think it was 5%, and it had the highest results outside of Keene. And remember at the beginning when I showed that first introduction picture, we were, assuming that flow through riffles was going to be one of the main drivers of our hyperreac exchange in these streams. And lo and behold, the stream with the almost no riffles has some of the highest. The Chester control, and Karen mentioned that, or Val mentioned that there's the degraded control. So that's a ditched wetland stream that is still in its straight ditched state. It has not been restored. It had the lowest of the three. Kind of maybe as you expect, we're calling it degraded. We say it doesn't look healthy, and it appears to have the lowest processing rate, lowest hyperreac exchange out of the three. The treatment was lower, and then the control was highest. There was some differences in the geomorphology of the stream between the control and the treatment there. The control was a forested, kind of cobble-bedded pool riffle stream. And then the treatment were these highly meandered wetland streams with deep pools that had a lot of cattail standing in them. So just the way they maybe respond was very different. Sargent had the lowest rates overall. It's a pretty small watershed, pretty small stream. The discharges were very low. But the treatment, and that actually had some of the best chloride data, so there's maybe the least amount of uncertainty in maybe the processing rates there, and the treatment's higher. Mission had the lowest quality conservative tracer chloride data, but the results are in kind of that similar order of magnitude as everything else, so the results look reasonable at this point. And they are pretty close to when we went back in 2023 to Mission Creek to try a different conservative tracer. So we'll go to the next one. So again, we're trying to maybe look for some things to explain the results we're seeing. So what this is showing here is that some just physical parameters of the stream velocity that was measured, stream sinuosity, variation in depth, how big grain sizes are in the pools, how big grain sizes are in the riffles, the reach slope and the percent riffle, and then the rate is what we're trying to correlate to. And what you can see is there's nothing really that has a great correlation. The best in this data set, and this is just for Mission, Sargent, and Chester. The best one is that percent riffle, kind of as expected. But some of the things is each of these streams is different, is a kind of a different geomorphology and different type of stream system. So if I added that Miller control on here, it would be at, if you look at the bottom x-axis, you can see it goes down like 30% percent riffle. Well, Miller would be at 5% riffle and have the highest processing rate. So this um, goes, this correlation completely goes away if I include that data set. So I don't really have enough data from these various types of streams to explain all these possible variables. Next slide. So conclusions based on the work from 2022 is that there's no real difference in the data that I've collected between the in-reach scale Razazaran processing rates and hyperreach exchange between treatment and control reaches. The restorations don't appear to result in lower hyperreach exchange based on this data. And at Chester Creek, at least, the treatment was better than the degraded control. So, and that's what the treatment reach looked like prior to restoration. So it appears that there is possible some improvement in that system, taking something that's a ditch and making a creek out of it. Looks like it's a good thing from hyperreach exchange point of view. Again, lots of different stream systems were sampled, but nothing was really sampled more than once. So I'm trying to compare a step pool channel to a wetland channel, and maybe that's not, maybe they're going to respond differently to restoration. So we would want to maybe compare wetland streams to wetland streams, and so on. Again, discharge has a strong impact on the hyperreach exchange and the processing rates. So trying to compare values from Keene that was done in June with 100 liters per second to Sargent that was done in late July at 3 liters per second. Maybe not. That's why we're trying to compare control and treatment. But again, maybe those differences change at different discharges between the control and treatment. And something that is important to note is by looking and sampling the surface water, we're looking at the bulk processing rate over the reach. We're not really looking to see if the restoration is impacting specific flow paths under the stream. You need to use subsurface sampling in order to actually see if you're changing the flow paths from restoration. So what I'm specifically looking at is the bulk over the reach, which has more cumulative impacts on the water quality and maybe temperatures if there's any buffering impacts with enough hyperreach exchange. And then lastly, chloride didn't work and it added a lot of uncertainty to my data. Thank you, Tom. He made a comment in the chat there. So some of the next steps is choices of conservative tracers. So this is what we went out back in last year that got the radio station out to watch us. We wanted to find something different. There's been 2 other conservative tracers used in the literature. One is fluorescein and one is bromide. Fluorescein sounded like a great alternative. I'm pretty sure I've heard that's what they use in Chicago River on St. Patty's Day to turn the river green. It's cheap. It fluoresces, so you could put it in the same lab equipment that you use to measure ras and ru, measure it at the same time. Keeps the cost down, moves fast. But we found out right before we used it that it is not stable in sunlight. It will degrade in sunlight. And then everybody in the literature, when I went back and read a lot closer, they run their experiments overnight, which we did not feel comfortable doing. The people who use it, they've got extra equipment that we don't, that allows them to do that. Other ones to use were bromide, which is again a salt. But we assumed that it was going to have low concentrations in our creeks. We went and took out some samples before we did the experiment and had them run on an ion chromatograph. And yes, they definitely have pretty much non-detectable levels of bromide at Mission Creek, but this still requires the use of an ion chromatograph, which increased the time and cost. But we decided to use the bromide. So we'll go to the next slide here. And this is the difference. So the top two rows are the mission control and treatment using chloride in 2022 versus using bromide in 2023. And these are just concentrations, changing concentrations of bromide over time and chloride over time. So you could see bromide basically comes back to zero like we would want. It goes up, it comes down. The data is fairly smooth. It's certainly way smoother than what we saw in the mission treatment or the chloride data. So bromide was a significant improvement. So moving forward, any of this work, bromide seems to be the way to go. And then I think next slide is kind of my last slide. So kind of where to take this next. This tracer system seems to show promise to determine hyperretic exchange in these North Shore streams. There's a lot of variables that impact hyperretic exchange, and stream geomorphology and stream system and valley type are probably some of the overarching controls. And so it'd be good to have more of this data in each of these systems to actually see like, okay, what is natural. Streams vary in time as well as space. So repeated experiments over the same reaches may show how it varies with discharge and whether or not these trends are consistent. So I have one data point for each stream and a control and treatment. Other studies that have tried to get at hyperretic exchange using only conservative tracers maybe have done 15 or 20 experiments on the same reach and then actually do like a statistical analysis on whether or not they're different. Obviously I wasn't able to do that in this study, but that's something you could see if there's actually statistical differences on the same stream of a given type over time. And lastly, the hypothesis that we're altering the subsurface when they're doing these channel realignments to actually get at that and highlight any differences in subsurface flow paths requires subsurface sampling. The current literature is done on sand and gravel streams. Shows that pretty much all of the rhizazurin decay occurs in the upper 2 to 5 centimeters of sediment where the microbes are living. So in depth, what that means is if they sample deeper than that, there was no rhizazurin left at 10 centimeters depth. Everything was converted to that daughter product, resveratrol, by that depth. But in our North Shore streams, our average grain sizes are generally significantly larger than 5 centimeters. So like the question is do our flow paths go significantly deeper given that higher permeability and larger grain sizes? Would that be different? That larger grain size also makes it very hard to devise a sampling system that you can get down through those large rocks, but you may be able to see any changes in the actual flow path depths with subsurface sampling by going that route. So yeah, I think that about wraps me up. I think Valerie
Okay,
might have one more
thanks.
slide.
Yeah, we have one more slide.
Yep.
So thanks, Brad. And I think didn't hear Brad mention this, but as far as we know, this is the first time that this resveratrol, resveratrol method has been used in Midwestern streams. And, so as Brad was saying, we ran into some of the interesting conundrums with using it in systems that have much larger bed size. Just a reminder that next year we will have more results. We're still finishing sampling the fish and macroinvertebrates and habitat and geomorphic stability in these systems, particularly the ones that just underwent restoration and realignment last summer, and then we're going in this coming summer to do that sampling. So with that, we should thank the literal army of folks who helped us get all of this sampling done. And those of you who've done tracer studies know that you go out early in the morning and often you sit there all day just collecting samples from the stream. And so Brad took a number of people with him. Every time he did that, it was the same thing for the nutrient cycling, had to have a whole bunch of people there all day taking samples. And, some of them were, okay, 12 hours, the chloride's still not down, it's still going up, and come back after 24 hours and see what happens. So all of these people helped us. I want to note in particular Dr. Ricardo Gonzalez Pinzon, who had came out to from University of New Mexico, who came out here and taught us this particular method to use. So with that, again, a thanks to our funders, LCCMR, and we can take questions. And I'll stop sharing so we have some hope of seeing the chat.
Hey, great. That was a really interesting presentation, Brad and Valerie and Karen. While people are thinking up questions to throw in the chat, I do have one that springs to mind immediately, and that is when you were doing the dye addition to the water, I couldn't help but wonder, did you have any concerned citizens like call in to emergency response saying, oh my God, something is happening to the creek, you better get out here right away? Or did you have any people that noticed the dye in the water and raised the alarm?
No, we did not. We did alert, thanks to Karen and Val's
Hmm.
suggestions. We did alert like the authorities. We let people know that we would inform the police. I think we let somebody at the PCA know, right, Karen?
Hmm.
We sent some
Yeah,
emails out.
PCA, fire department, Coast Guard. But what we realized too is that, yeah, it looks really impressive right when you dump it in. 100 meters downstream,
Hmm.
you can barely see
Hmm.
it in most streams. And so people would have had to been like right there with us to really notice it.
Well, I do have one question. Having worked with Rosazurin a little bit in the past, I know it's a real sensitive indicator of redox status of water.
Mhm.
And I'm wondering if what you might do is correlate those, take dissolved organic carbon samples of your water and see if that correlates to your level of Rosazurin and the transformation product, because it might be a proxy of your bacterial activity that
Yeah.
is transforming that, right? Because I think that the DOC and would be a good indicator of how much microbial activity you might have in those upper sediments. It's just a kind of off the top of my head thought. I kind of wonder how you might think of that.
Yeah, that is interesting. I mean, yeah, it is exactly what it is. It's directly measuring microbial activity. We're just kind of using and saying that, okay, the microbes are on the sediment and the subsurface. Therefore, if it's transforming, there is some if you're, how does this work? You can have a transport limited system or a reaction limited system. And if there were enough, if the flows through the hypereoic zone were slow enough that you would be getting 100% transformate. You could tell if you get the right parameters, if you get a good transient storage model, that is one thing that you can look at. If you have a reaction limited zone or transport limited zone, as I mentioned, we didn't really trust anything with the transient storage models with this data set, and kind of are jumping that we're basically in transport limited systems. Everything's moving through fast enough that it's more of the how much water is getting in and out as opposed to whether or not the bugs are being overwhelmed. Not bugs, bacteria are being overwhelmed.
I think it would be interesting. We might be able to do some comparisons between what we were seeing in the nutrient tracer studies to what you found for these different creeks with the processing of resru, and just see if there's something there. Because as you mentioned, we've got a wetland stream, well, a couple of wetland reaches in there that may not be, really comparable very well to our standard riffle run of a cobble bed stream.
What seemed encouraging to me was that at the end of the study, the restored regions or the treatment areas of the stream seemed to have the same results as the control portions. At least that's what I took away from it. Is that the right way to look at this? And I would think that that's encouraging news, that these restorations are restoring more than just flow path, right?
Yeah, I would say based on this data set, there's no discernible difference between the control and treatment in this specific regard. Yeah, the hyperrig exchange, they're not, I wouldn't say they're making it better than adjacent reaches, but they're not making it worse either. It's kind of a neutral result. Based on this data set, like I said, there's a lot of caveats on that, but with this, yeah, that's what I would say is that it's not making it worse.
So
Or
Jesse Anderson,
hyperretic exchange.
yeah. So Jesse Anderson typed something in the chat. Great work. Do you have any thoughts on the impact of the 2012 flood of record on hydrologic transport in these streams based on your research?
Karen, I
I
don't know, this
mean,
one might be for you.
yeah, I think it's you, Karen.
I mean, the short answer is I don't think we have the ability to tell that based off of what we're working on, in part because we don't have any data from before the flood. But also, yeah, I think that would be getting at whether you have like more subsurface storage or more surface storage or changes in how the water is moving through the system. And I don't think we have the ability to answer that question.
Tom asks, do you know if the restoration design or materials used for the bed is installed with hyperarray connectivity in mind or being maintained?
I mean, I can attempt this one as well.
Yeah,
And so
please.
the short answer would be we do not know this, but for most of the restoration designs that I've seen around here, hyperreach connectivity is not a specific design parameter. Connectivity in general is always a goal, but it's usually looking at longitudinal connectivity and connectivity to the floodplain. And generally the bed would be designed to sort of match a reference reach, a geomorphic reference reach in terms of grain size, et cetera, and then think size so that they are not going to blow out, so to speak. So I have seen places where hyperreach connectivity is something that's being designed for, and nutrient uptake is something that's being designed for. And there's some cool work going on out like in Chesapeake Bay, where nutrient loading is a really big problem. And so if the restorations there can address specifically like nutrient transformations, that's a really good thing.
Now, Jesse asks, did you notice any differences due to the geologic conditions? Clay-dominated geology at low elevations versus gravel-dominated areas at higher elevations?
Yeah, so ours is backwards in Duluth. Kind of. We have more clay on top in those wetland systems in some ways. Depends on the area. I would say, did I see any differences related to that? Maybe. Like I said, I don't have enough data points, but each system I did was different. So like Mission was in the alluvial reach with pretty coarse substrate near the mouth. Chester and Miller are on top of the hill in the wetlands with very, very fine substrate, muck and clays. And then Sargent is interesting. That one was in lacustrine clays. It had very low discharge, but it also had very low processing rates. Like the bed itself had, in the control reach, had fairly coarse grains. However, the walls of the valley are, lacustrine clays, so pretty tight. So yeah, I was really hoping to try to see any differences between the general geology of the area on some of this and whether or not it have an impact. Like, Mission is in this lacustrine clays, but it's cutting down into sandstone bedrock. And I was hoping we would get to keep East Beaver because that reaches on volcanic bedrock under the alluvial sediments. So I was wondering if we would see any differences there. Unfortunately, we need to test that idea. So it's interesting idea, and I think that'd be really cool to tackle with some of the subsurface sampling if you could get after it.
Hmm.
But like if we went and did East Beaver now that it blew out, we could maybe answer that question because
Hmm.
all of that picture that Val showed earlier of East Beaver with all the large cobbles and boulders in it is all gone, and now the creek bed in what was the restored zone is now right on lacustrine clay all the way through. So I would expect to see very minimal exchange there because it's kind of like a water slide.
It is a water slide if you try to walk in it.
It is.
Holy moly.
Yes.
Huh.
Um,
Huh.
and yes, so yeah, we forgot to mention we did ask to see if we needed permits before we dumped dye and chloride and bromide in the stream. So we were working with Tom on that one. So yes, we did check with y'all to see if we needed permits for this.
Well, we are really right at time for those people who have to jump off. Hey, this has been a great presentation, and thanks for agreeing to come tell us about your work. It sounds like just really interesting stuff, and I kind of assuming you're going to be continuing this work going down the road.
Well, we're kind of grant funding limited, so if you
Okay.
hear, we would love to continue and especially to visit, we're we feel like the streams that were just restored last summer if realigned last summer aren't going to have much of a chance to recover before we have to sample them this summer, but because of the length of our grant, our funding, this is the latest summer that we can sample them. We would love to get a chance to go back in later on and sample them again, give them what is considered a fairer shake in looking at how well these restorations did, but we need funding for that. So
Hmm.
we are a bit limited there.
Ah, well, great. Hey, thanks again for coming to talk to us. It's been just really interesting. So thanks everybody for coming, and our next talk will be in a couple of weeks, I think. So see you then.
All right. Thank you.
Thanks for having us.
Yep. Bye-bye.